Dunning, who commenced her tertiary career in 1998 and received her Bachelor of Science with honours in 2001. In 2002, she enrolled in the School of Molecular Biosciences, discipline of genetics, at the University of Adelaide. And then between 2004 and 2008, she pursued further studies in the disciplines of obstetrics, gynecology, and became Dr. Kylie Dunning, Doctor of Philosophy in those disciplines in October 2008. Kylie has given a great deal of her time to her chosen profession and to the community. Has received many awards and prizes, has sought and received substantial research funding, has sought and been successful with significant scholarships and travel awards, and has been invited to give presentations both here in Australia and overseas, in New Zealand and the United States, and has been very heavily involved in research training. Her CV actually covers some 3 A4 sheets, so I'll let her tell you the rest of the story. To speak on the subject of cancer research and therapies, please make very welcome our guest speaker today, Dr. Kylie Dunning.
Thanks for having me, and what I'm going to talk to you about today is a very small part of some of the research that I do.
Yeah, it's working.
Oh. So a cancer patient's greatest concern is will I survive? But survival rates in Australia have improved dramatically over the last 3 decades, and now 80% of young people who receive treatment survive. So as a result, life after cancer is becoming a reality for many people, and in doing so they're able to plan for the future. And in planning for the future, the second greatest concern for these patients is, will I be able to have children? Now, not that long ago, the ability to have a family after cancer would have been a fantasy for most patients, but now our work is helping to make this happen. So you're probably aware I'm going to be talking to you today about fertility preservation for cancer patients. So options for male patients have long been available, with the ability to collect and store sperm for use later in life. And a most famous example of this is Lance Armstrong. All of his children were conceived using sperm that was collected and stored prior to him receiving treatment for his cancer. However, unfortunately for female patients, there are very few effective options for her to preserve her eggs. So the goal of our research is to help women diagnosed with cancer preserve their fertility. And we now know a lot more about how to grow healthy eggs outside of the body to help make this happen. But before I get to that, let me first tell you a little bit more about the problem. So based on Australian statistics, 1 in 3 people will be diagnosed with cancer at some time in their life. And while cancer is often thought of as a disease associated with aging, it also affects young women of reproductive age and girls. And one of the side effects associated with life-saving chemo and radiation therapy is that it can also reach the ovary and pose a threat to fertility. But by initially keeping the egg safe while the patient undergoes treatment, we have now made a major advancement in understanding how to best grow eggs and embryos outside of the body, so that later in life, when she's ready to have a family, in the future we'd be able to put these embryos back in order to form a pregnancy and a healthy baby. So how does cancer treatment affect female fertility? Well, before I get to that, I'll first give you a quick crash course in female reproductive biology. So this is a diagram of an ovary, and this is where all the eggs are found. You can see the eggs shown there in yellow. And around each egg are cells that grow and develop around the egg. And the role of these cells is to support and nourish the egg as it grows and develops in the ovary. And together, the egg and these cells make up what's called the follicle. Now, a woman's ovary contains many thousands of follicles, and you can see here, if I can get the point to work or not, you can see follicles at different stages of development, from a smaller follicle on the left at the top to a larger follicle on the right. So women are born with all the eggs they'll ever have, unlike men who continue to generate sperm throughout their life. And one of the side effects is that cancer therapies can damage these eggs. So I'll now go on and talk to you you a little bit more about why the eggs are so sensitive. So cancer treatments like chemo and radiation therapy also reach and affect the ovary, and this can lead to the death of all the eggs in the ovary. This can result in premature menopause and infertility. So how can we best protect these eggs and preserve the fertility of these women? So one option is ovarian cryopreservation, and this process involves removing a slice of the ovary. This tissue can then be frozen, and then later in life, this tissue can be thawed and resuscitated in order to restore fertility. So the goal of our research is to understand the best way to grow eggs and follicles outside of the body in the laboratory in order that they can produce eggs of a certain stage that can be fertilised by sperm and develop into an embryo, similar to how traditional IVF works. So I've been working on an exciting new way to grow follicles in the lab. And this involves, but to do this research, we first need to obtain follicles. And to do this, like most scientists, we first work on mice before we work with human tissue. So this new technique that's been developed to grow follicles in the lab involves isolating individual follicles. And then what we do is that we encapsulate these follicles in a three-dimensional ball of gel made of a substance called alginate. Now, alginate is a natural and safe product. It's derived from seaweed, and it's already found in many of our foods. So we then grow these follicles in the alginate ball, and we grow them in vitro, which just means that they're grown in a dish, and they're grown in warm fluid that replicates what's normally inside the mother. And because these alginate gel balls are transparent, we're able to watch how this follicle grows and develops over time in the dish. And so you can see here, this is a follicle suspended within an alginate ball. And if we take a closer look, you can see the edge of the alginate ball on the left-hand side is a transparent edge. And you can see the follicle in the middle with the oocyte, the round oocyte in the middle. I can't really show it, sorry. So the great thing about this technology that's been developed is that it recapitulates how the egg and the follicle normally grows inside the body in the ovary, such that at the completion of follicle growth, you can see the egg contained within the middle of the follicle, as normally occurs in the follicle in the ovary, shown in the bottom. We see the development of a specialised layer of cells that directly surround the oocyte, called cumulus cells, and this normally occurs in the ovary also. And finally, we also see the development of this fluid-filled cavity, which is shown in that photo in the middle as that clearing of cells, and this fluid-filled cavity also occurs in the ovary, in the body, and is required for ovulation. So the other great thing about this system is that at the end of growth in vitro, I'm able to extract those eggs, fertilise them in vitro with sperm, and then the resultant embryos, we're also able to watch how they grow and develop over time in the dish. So we can watch how these embryos divide into a 2-cell embryo. 2 days later, a day later, they develop into a 4-cell embryo. A day later, into an 8-cell embryo. And these embryos continue to grow and divide over time until finally they get a little bit larger, and they develop that large cavity in the centre. And it's at this stage of development that the embryo is able to implant into the womb and form a pregnancy. So this technology is really great, but we wanted to take it one step further. So we wanted to know whether we could improve this therapy, making it a more viable option for patients in preserving their fertility. So to do this, I looked at what the egg requires in order to be healthy. And you require healthy eggs because only the healthy eggs are able to be fertilised by sperm and develop into an embryo. And so we know from the work of other scientists that how much energy the egg and the embryo, the egg and the follicle, are able to generate determines how healthy the egg is. And we also know that if we burn or metabolise fats, we're able to generate more energy than if we burn or metabolise sugars. So based on this knowledge, I asked, if we increase fat metabolism by the egg and the follicle, can we obtain healthier eggs? So if a cell is to turn or metabolize fat into energy, the fat must first enter the power station of the cell. And this is called mitochondria. And this transport is dependent on a molecule called carnitine. So carnitine is attached to the fat molecule, and this enables its entry into the mitochondria. So you can really liken carnitine to being the train which gets the coal, or in this case the fat, to the power station. So carnitine is a naturally occurring molecule. We normally obtain it from our diet, and it's normally found circulating throughout the body. And it's absolutely required if your cells are required to turn fat into energy. So based on this, I next asked, if we add carnitine to follicle during 3D growth in vitro, can it increase fat metabolism and increase the number of eggs and embryos produced? So to answer these research questions, I isolated individual follicles from ovaries. These were then grown in the three-dimensional alginate system, and these were grown under our standard normal conditions. But to test what effect carnitine would have, we isolated separate follicles. Again, we grew them in the three-dimensional alginate system over time, but these follicles were provided with carnitine. So we first looked at what effect carnitine had on the ability of the eggs and the follicles to burn or metabolize fats. And so what we found was when follicles were grown in the presence of carnitine, follicles were able to burn 30% more fat than if they were grown under our standard normal conditions. But what effect this had on the egg, we next looked at. So we looked at whether carnitine had an effect on the ability of follicles to grow mature eggs. So that's a photo of a mature egg, and you need a mature egg in order to be fertilised. So follicles grown in the presence of carnitine produce 20% more mature eggs than those grown in its absence. So we next looked at how healthy these eggs were. And so we do this by looking at their ability to be fertilised by sperm, and also their ability to develop into an embryo like the one I showed you in that video earlier. So we found that eggs derived from follicles grown with carnitine produce more than double the number of eggs capable of being fertilised by sperm. And excitingly, these embryos went on to form 3 times more the number of healthy embryos than eggs derived from follicles grown under our normal conditions. So for the first time, we've shown that fats are a vital energy source for the egg and the follicle, and that the addition of carnitine during their in vitro growth resulted in increased fat metabolism and increased energy production. This in turn resulted in more mature eggs being grown. More of these eggs were able to be fertilised by sperm, and these resulted in 3 times more the number of healthy embryos. So we've gone on to extend this work, which is done in the mouse, to see what effect this would have on human follicles. And this work is done by our colleagues in the US. So these researchers have access to portions of human ovarian tissue that's been donated by cancer patients. And you can see here the growth of a single human follicle, which is grown in the three-dimensional alginate system over 14 days. So even in initial experiments where carnitine was added to these human follicles grew and developed better than they ever have before, making it a great advancement in the therapy. So in advancing fertility preservation for cancer patients, we've found a way to grow healthier eggs outside the body in the lab. And this resulted in healthier eggs and healthy embryos. So in the future, we'll be able to transfer these embryos back into the patient in order to form a pregnancy and restore fertility. So what does this mean? Well, what this means is that we've provided new hope for cancer patients, hope in restoring their fertility and having a family. And this work forms a part of the research interests of the institute called the Robertson Institute, of which I'm a part of. So the Robertson Institute brings together over 350 researchers and clinicians, and our research areas include the health of women and babies, early origins of disease, reproductive health, and stem cell research. We're also based at many different locations, including the University of Adelaide and and all the teaching hospitals. So we also have a foundation, and the role of the foundation is for the community to be involved and engage and support our research, and also to raise funds to grow new areas of innovative research. So this will enable us to find new cures and treatments that will provide long-term health outcomes for patients. We're also very fortunate to be supported by the Governor of South Australia, His Excellency Rear Admiral Kevin Scarce, and his wife Liz Scarce, who are joint patrons of our foundation. So if you'd like more information about other research we do. If you'd like to chat to me about other research that I do, I'd be more than willing to chat. Or you can visit the website or take home one of our annual reports or Foundation newsletters. So thank you very much for having me today and let me share this with you.
Thanks very much, Kylie. Kylie's very happy to answer questions, so the floor's open for questions.
Kylie, in the bulletin, it talks about the work with migratory birds. How did you connect A with B?
It's really just it was a way of illustrating how it's difficult to interpret how a tiny cell like an egg would burn energy. So it was really just an analogy to show that migratory birds will store fat and burn fat over long periods of time and then, to generate energy. So our work is similar in that these tiny cells are also able to do the same. So they have stored fat within them, as well as fat that's supplied externally. So it's simply an analogy.
Right. I wasn't that perceptive.
Not into birds. Yes, Ralph.
My wife had no difficulty in conceiving. We had 3 children, but the third was the last, because she was always had morning sickness. What have you done to relieve pregnant women with morning sickness?
It's a little out of my field. However, I believe there are some medications that women are provided now. They're similar to anti-nausea medications that people receive in cancer treatments, and they have been proven safe in pregnant women. But I don't, it's not really something, it's a natural part of pregnancy, and it's difficult, but it's not my area of expertise. I'll be more than happy to follow it up and get back to you if you like.
Kylie, thanks. That was extremely interesting. Now, what you've developed is a solution to a problem that is created by a solution, which is chemotherapy. Is the Robinson Institute or similar bodies involved in something that removes this very effective, long-standing but damaging treatment?
So this is just one tactic in order to preserve fertility. There's many options available. This kind of option is really applicable to women who don't have a partner and also prepubertal girls. So the issue is that the best way we have to preserve fertility at the moment is to give patients an Ivf cycle, so to stimulate their ovaries, collect mature eggs, fertilise them with sperm, and then we're able to freeze the embryos. We're not at the moment able to freeze mature eggs because they're very similar to lettuce. You put them in the freezer, they're full of water, they crack. They just don't do very well. So this kind of treatment is very applicable to certain patients. That if you have someone who already has a long-term partner, the Ivf cycle and embryo cryopreservation is a good way. Another tactic that other researchers are working on is looking at down-regulating. There's already treatments available to down-regulate their reproductive cycle, so to put the ovaries in a sort of dormant state, so that the more dormant they're in, the less susceptible they are to chemotherapy. And then there's the other, the younger you are, the more eggs you have, and the less likely you are to be affected by chemotherapy. Whereas if you're a bit older, you have fewer eggs in reserve and they're more susceptible to being damaged. And there is other research going on looking at how to protect the ovary from chemotherapy. And there is another thing, so if you're having directed radiation, they can also move the ovary. So they'll cut it from where it is, transpose it to another part of the body where the directed radiation therapy isn't occurring. So there's a lot of research going on, and this is one part to help a certain set of population.
Frank O'Neill.
Thanks, Kylie, for a very interesting insight into the development of science and technology. It raises some issues for me, the issue of managing fertility outside the body, if you like. It raises a range of ethical issues. Who makes the decisions about who can have it? Are we getting to a stage in our development where we can create new babies that can be bought by others, as pets perhaps,
personally am a basic scientist, so I'm really interested in, how the follicle and the ovary grows and matures an egg. And this is the kind of application it would have. But however, my research to be translated clinically is most likely done by somebody else in another country. So that's what's happening at the moment. There are a lot of committees with senior researchers and people from the community who will debate and decide what happens, what's allowed to be happening. And does that answer your question? There's a lot of legislation, and it's a difficult one to answer. Would you like me to follow anything else up? No?
I understand that as a scientist, you're just doing science, and you will develop technology, but at the end of the day, technology will be developed, it will be used. How it will be used will require more judgment. And
It's very true.
I'm not sure that we've got that right in our society.
Well, at the moment, in Australia, you can't be given money for donating an egg or sperm, unlike other countries. Other countries you can get paid for donating gametes, so your egg or your sperm. So here in Australia, it's, you can only do it out of the goodness of your heart. So I think that's a good start in Australia. Yeah, don't get money for giving blood.
Hi, Kylie. Thank you again for addressing us. My question is, what makes sperm different from eggs or from a human being? So going back to science, and to be able to be frozen and to survive, years in
Sure.
that frozen state.
It's really about how they're packaged up. So sperm are really packaged up small. So the sperm is much, much smaller than the egg. So all its DNA is wrapped really, really tightly. It has certain mechanisms in order to survive in the environment in which it has to live and survive before it reaches and fertilises the egg. It's just a very different process. So the egg is much, much larger. There's debates on why the egg is larger. So is it to hold enough energy to maintain embryo development until the embryo implants into the womb and establishes a blood supply? But the major thing about the cryopreservation is that it has a high water content. So that it just doesn't, it will freeze okay, but when you thaw it, it will form ice crystals. And we just haven't got the cryobiology yet that's sufficient to maintain them. We're very good at freezing many other cells, just not very good at oocytes yet. And they're the largest cell in your body. You can see it by eye.
One more question.
Yes, thank you very much for your talk. I'm interested in two quick questions. One, are you actually applying this new technology now? And secondly, does the Robinson Foundation gain through the sale of its intellectual property? How does that actually provide additional revenue? Because I presume this technology could be used worldwide.
It depends on whether the technology is able to be patented, I can't think of say the word. So if you're able to put a patent on your technology, the thing is that the University of Adelaide owns your IP. So you'll get a portion of the funds back, but a lot of it is, a lot of the IP funding goes to the university and the department, not the individual per se. Does that make sense?
Sure.
The foundation works differently, so it is its own, you can donate money, and if you wish that it goes to a certain area, it can be directed to that area of research. So if you're more interested in your funding your stem cell research, it's very different to how the money from intellectual property, so if you're able to translate it and a company purchases the IP, it's very different. This work wasn't able to be patented, so I won't be making money off of it or anyone else, but we are working with other people, so we're working at looking at what effects it has on other species, like cow. Our collaborators in the US are working very heavily on what effect it has on human follicles. So for me, it's about the discovery. It's not about the dollars you make at the end of the day. It's the excitement of the discovery.
Kylie, on behalf of everybody, thank you very much for a most entertaining and instructive talk. I have to say that for
for one so young, one wonders where you're going to be in 20 years' time. But well done, congratulations, and this club always gives the guest speaker a gift. It's not a glass, it's not a photograph of yourself, it's not an egg cup.